Okay.
There we go. Okay.
Everybody, thank you very much for coming to this meeting today. I really appreciate it. This is an exciting study. It has been a long time coming. It addresses a long-standing question in burn shock resuscitation. And we have recently enrolled our first patient here at the U.S. Army Burn Center in San Antonio, Texas, and we look forward with gratitude to your participation. Next slide. This is our agenda for today. Next slide. So the clinical sites are our burn center here, UAB, you folks at Vanderbilt, folks in Washington State, UT Southwestern in Dallas, and UTMB in Galveston. There is also a data coordinating center at the University of Washington, and we're sending samples to the University of Maryland and to the company that is responsible for the pathogen-reduced plasma, Cirrus. The coordinating center is led by Monica Phillips on this call. It's the Coalition for National Trauma Research, previously known as the National Trauma Institute, and it's here in San Antonio, Texas. This is the sort of timeline of events, and as you can see, we are well into the period of performance of this study. So we are anxious to be enrolling patients into the study at this time. Next slide. So the hypothesis here is that the administration of plasma for burn shock resuscitation will reduce 24-hour and 48-hour total fluid resuscitation volumes, as well as the incidence of ARDS, multiple system organ failure, and any other resuscitation-related morbidities. Specific aims address the fluid resuscitation volumes, number 2, ARDS, and number 3, some measures of the so-called endotheliopathy of burn injury. Next slide. This is an open label phase 4 multi-center randomized controlled prospective clinical trial. And we have 2 groups. Next slide. So the primary endpoint is the total volume of resuscitation fluids, all of them, whether crystalloid or colloid, between hours 0 and 24 as measured in milliliters per kilogram. Next slide. We're also going to look at the first 48 hours and the time frame between 0 and 24 and 0 and 48, normalized in the usual way to mLs per kilogram per percent burn. We're going to measure reference as an index of hemodynamic instability. We're going to measure arterial lactate levels, and we're going to look at rescue. We define rescue as being the initiation of extracorporeal therapy like CRRT during the first 48 hours, the infusion of high-dose ascorbic acid, and or the initiation of a continuous infusion of albumin during the first 24 hours. And I would like to emphasize that, for example, in our practice days, it is fairly common here for us to use 5% albumin before the 24th hour post-burn. We would consider this a rescue intervention, and we will measure that. We do not wish to prescribe that, prevent it. We leave that up to the clinician. Next slide. We're also
Can I interrupt for just 1 second on that last slide?
Yep.
You say albumin before 24 hours. I know in the study meetings we talked about how are we going to treat anyone that gets albumin in the first 10 hours? Like, would that disqualify them, or is that just going to be part of the study? Have we come to a decision about that?
Yeah, Monica, do I owe a memo on that topic?
Yes, you do.
Can you remind me? Can you send me an email to remind me
Yes.
Thanks.
Mhm.
So if the patient is receiving a continuous infusion of albumin at the time of study enrollment, right? It is as if the patient is already being rescued. So, for example, if the patient comes in and is considered for enrollment, of course, before the eighth post-burn hour, but is already receiving a continuous infusion of albumin, we would consider that patient excluded on that basis.
Okay, so we have
Because-
decided that we were going to exclude, because the way the protocol was written was like any continuous infusion of albumin before hour 10 would
Yeah,
exclude.
I think we need to change that to hour 8. That's what we decided, right?
I
mean,
think that's what was
be-
discussed at the meeting, but
Yeah. And that's my recollection as well. Just send me an email so I make sure I clarify that in writing. And the
I will.
reason for that, criterion, excuse me, the reason for the exclusion criteria is that we are considering the initiation of a continuous infusion as evidence of rescue, right? So if the patient's already being rescued before that we enroll them in the study, it does not permit us to assess that secondary endpoint. But we didn't want to say, we decided the 10-hour concept was flawed.
Okay.
Anyway, these are the secondary endpoints. ARDS, mechanical ventilation, ventilator-free days, SOFA scores, ICU in hospital, length of stay, in-hospital mortality. We're going to look at some patient-reported outcomes, and we're also going to look at trolley and VTE events. Next slide. These are our measures of endotheliopathy and inflammation and thrombin generation. Next slide. These are the inclusion criteria. Burn size needs to be 20% or greater. And the most important point is that the patient needs to be enrollable within 8 hours of injury, not of burn center arrival, but within 8 hours of injury. And that's going to be, that's really the major sort of challenge in a study of this nature. Next slide. These are exclusion criteria, and they include non-thermal injuries and sort of end-of-life care, individuals less than 18 or over 65 years of age. And we talked about the albumin or other rescue procedures would exclude a patient. Some indices of severe pre-morbid disease as well. Next slide.
Can I clarify? We've been talking about continuous albumin infusion.
Right.
Do we treat like if someone gets, 50 ml of albumin or something at some point, does that count?
Yeah, I mean, in my opinion, the answer is we're not count that. Like the patient comes in, let's say they get a bolus of a narcotic, hypotensive, and they get some 25% albumin. Probably happens pretty frequently. We're not going to count that.
We're not going to count that as an exclusion criteria.
Correct.
Right. Okay.
Thank you. Next slide. So in terms of informed consent, we are able to use a legally authorized representative or LAR to provide consent. And we are able to do e-consent through REDCap. Now, what we found during our enrollment the other day was that this whole process could be accomplished using an iPhone, using a smartphone. We had the steps that we took were as follows. Step one, we became aware of an inbound patient, and we got the patients through our dispatcher. We got the patient's next of kin phone number. A physician from our team called the next of kin and told them that their relative was coming to the burn center, told them in general terms about the study, and told them that a research coordinator would be back in touch shortly. We had our research coordinator follow up with another phone call, and that enabled our CRC to send them the link to the e-consent. They're able to review the e-consent and sign the e-consent using the smartphone. No need for faxing paper, no need for a computer. We did it all through the smartphone. Monica, do you have any feedback on that process?
Nothing specific. It seemed to go well. I mean, I've done it on my phone to myself as a test, and it works pretty well.
It went exceedingly well, according to Joel, and just goes to show you that we live in a different era. And so I was quite pleased with that. Next slide. Randomization will be provided via the REDCap Data Coordinating Center. It's randomized according to TBSA, 3 different TBSA categories. Now, we decided that we would, if we mistakenly, assessed burn size such that the patient was in one category and then later reassessed burn size such that the patient actually belonged or should have been in another, that we would not change, we would not go back in the REDCap and try to reassign the patient, if you will. We would follow a sort of intent to treat philosophy in this regard. What if the patient's burn size is initially assessed as 20% or greater, but then later gets reassessed as being less than 20%? We would not, we would keep that patient in the study.
Can I ask, in those cases, do you reset their volume based on the correct TBSA?
Yes. And the Burn Navigator permits that in that software. By the way, another problem that we have had, and we've encountered that problem in the past when using Burn Navigator, and we have adjusted the burn size in the Burn Navigator software. And the other problem that comes up is when you set the weight initially, right? When you actually measure the patient's weight, you have to go back in and correct the weight. And Burn Navigator permits both those changes and also records the fact that the change was made by somebody in the notes section of the software in Burn Navigator. Next slide. This is the sort of strategy for resuscitating the patients. These patients are all started out on lactated ringers according to a rule which puts resuscitation volumes in between the 2 and the 4 ml per kilogram per percent burn range, kind of closer to 2. And that's called the ISR rule of tens, wherein you take 10, multiply it by the TBSA, and that's your hourly rate, and you adjust from there. That fluid is titratable. And then in addition to that, one group of patients gets crystalloid at a dose, additional crystalloid at a dose of 1 ml per kilogram per percent burn. Do not rate, and the other group of patients gets plasma at the same volume, 1 ml per kilogram per percent burn. The latest update of Burn Navigator does these calculations for you, for which I am very grateful. It is specific to this study. I don't know if you guys got updated Burn Navigator software, but it's
We do. We have the Propolis like update for our package.
often awesome. And then the LR, the LR number 2 or plasma are both stopped at 24 hours post burn. Next slide.
Can I ask a question that one of my faculty asked about
Yes,
this?
sir.
Can you go back to the previous slide, Monica?
Yeah.
Thank you. So our resuscitation right now is somewhat similar to the experimental group in that it is a rule of 10s LR resuscitation with a shorter but continuous run of plasma inside of it. So it's not exactly the experimental arm, but our question was more about, so on top, we have, for patients over 80 kilograms, we have a little offset so that we don't over resuscitate them that accounts for like every 10, additional kilograms, it alters it. So one of the questions that one of my partners had was, since the standard resuscitation, A, doesn't have plasma at all, and B, is different in that it doesn't account for the patients that are, over 80 kilograms. Is the consent written in such a way that it allows us to explain that they are like still an experimental group? They're not getting the standard as defined by our protocol. Steve, is that roughly your question?
Yeah, it's mainly that this standard or control group doesn't match our actual written standardized resuscitation for our burn center. And so the concern would be, if I'm going to do something that's off of our standard of care, even though I understand that there's equipoise around the different resuscitation strategies, I have to either consent the patient for that and tell them we don't know if our strategy is superior to the one listed here, but I am taking you off of our standard of care for this region, so that because many of these people will be big burns and they're likely to have bad outcomes just at baseline because they're big burns. If somebody comes back and says, we think that you started your resuscitation lower than you normally would have, and you didn't give FFP like you normally would have as a standard in your center, and then my person got AKI and ended up dying, right? Like, I can't say that that was okay on, like, if I'm in a lawsuit or something. So we have to consent against that.
Yeah, I mean, for your center, you need to be able to, you need to feel comfortable with the conversation you're having with your patients. I agree. Personally, I feel comfortable having, enrolling patients in this study in terms of having clinical equipoise, and also because the study provides the clinician with the ability to alter the resuscitation strategy based on clinical judgment, broadly stated. So at no time do I intend to sort of tie clinicians' hands, understanding that fact introduces the potential for variability in practice. And, I do anticipate variability in practice. Now, concerning the consent issue, the real question I have here is, you have a standard of care, but you also feel that there is probably clinical equipoise. So when you say there's a standard, it doesn't mean, in my opinion, that that it is the best therapy available, rather that it is your usual practice. So the question I have is, what exactly, it's kind of like a philosophical question, is what exactly do we mean by standard of care? Because it's known to be best, or because it is the way that we have decided to practice based on personal experience, best available evidence. But, bottom line is, I think this is a question that you would need to reflect on in your personal practice as well as a burn center.
So to follow up, and I know we've talked about some of these things already. So if I did have a patient that came in and I enrolled them into this trial and they randomized to control,
Mhm.
and I start them at this rate and then immediately titrate their rate to what our standard is, would they get excluded from the trial?
No.
Okay.
Oh, by the way, you raised an important point that I wanted to highlight, and that is that the ISR rule of 10s, 10 mLs times TBSA is the initial rate. You are absolutely correct. Goes on to say, for patients in excess of 80 kilograms, add additional fluid, etc.
Mhm.
And I concede that for the sake of simplicity, I did not include that extra fluid based on increased body weight, and balanced kind of against the fact that they're getting additional fluid in either arm in the form of either LR or plasma.
Yeah, my interpretation of this has been that like both of these groups, experimental, which is admittedly closer to our actual resuscitation, and the standard crystalloid resuscitation are like, you must be consented into them. And the consent does not say that one is the standard of care or a gold standard, because no gold standard, so to speak, exists, which is actually, the point of the study, right? Like, so as long as part of the consent conversation, we explain that, like, while there is, equipoise, I mean, you have to, I guess, make that into language that is understandable to the layperson. But as long as there is equipoise between these two groups, neither is the like resuscitation that it's not the standard resuscitation. They're both part of the experimental model, even though one is not experimental fluid. But like, that's a thing that I feel comfortable having as a part of the conversation in the consent conversation, and then enrolling with some confidence in these groups. Like, yes, we moved to a protocol that has, plasma in it and does account for over 80 kilograms because we believe that to be true, but that's not because we have any specific evidence to suggest that it's true.
And then if I enroll a patient and I give them FFP, that does exclude them, or did I misinterpret that?
No, once you enroll a patient, if you decide to start a colloid, we would continue the patient's involvement in the study, but we would count that as a rescue procedure. And that would be the case regardless of the arm the patient was in, crystalloid arm or plasma arm. If you will, additional colloid, we would count that as a rescue, but we would not exclude the patient. Okay. Although we would ask that you document why you're doing it, what led you to do it, this type of thing.
But if they got plasma for whatever reason prior to enrollment, that would exclude them theoretically, right?
I did not think of a scenario in which a patient would come in and just get a plasma transfusion, unless, for example, they were bleeding, in which case they should be excluded.
We have one center in particular that's a pretty high level trauma center already, and they do. So they'll take the way our referral pattern goes for them is the patients are in the community, they get burned, they bounce to there, they get their kind of initial stuff started there, and they will give FFP, and then they'll send them to us as the burn center. They used to be a burn center, so they know what they're doing, they just don't have the resources anymore.
Yeah, that's interesting. My inclination would be to exclude those patients.
Okay.
I think it's an exclusion criteria,
yeah,
that if they have received, yes, that if they've received plasma prior to, they are Excluded. Already receiving plasma infusion.
Yeah, so pre-enrollment, I think it is, that is
Correct.
the plan.
Yeah.
Yeah, so what happens if they just get a rapid plasma bolus at the referring trauma center? Not something that I had thought of,
Right.
but we're going to exclude that. Monica, next slide. A little bit about this pathogen-reduced plasma. It's basically another way of processing plasma to try to eliminate pathogens. Next slide. Point is that this plasma is FDA approved. And so this is not, for example, an IND type of study. Next slide. It is extensively approved by various regulatory bodies in the United States and around the world. Next slide. The blood bank is going to track the distribution of these units of plasma via REDCap. And it lasts for 12 months. Next, and of course, the volume in these bags is going to vary somewhat. Next slide. So the problem that we ran into with our first patient was it was after hours for the blood bank, meaning it was 4 p.m. or something. And the tech there in the blood bank was not aware of this study. And so, you know, we had to call the boss, and we got that taken care of pretty quickly. These units are transfused like any kind of plasma unit. Next slide. And we set up a special order set in our EMR for these plasmas.
That's the case with us too. I'm speaking to my faculty now, but yeah, that's like, we'll have a specific order set, and then our liaison from the blood bank is Dr. Jenna Andrews.
Roger. So these are labs, and the labs are going to be drawn on admission, and then 12, 24, and 48 hours post burn. And so Monica, do they have kits?
They're being sent out today, actually.
Okay, awesome. So that you guys will receive kits with tubes and instructions in them. Monica, anything else you want to say about that?
There is, we'll go through the training at the end, but there is specific training as to how to centrifuge, et cetera. But if your staff would need it, if they haven't done it in a while, there's actually a little video of somebody performing the centrifuge and then the aliquoting, et cetera. So all that's available on the website.
All in all, I think it's pretty straightforward, basic stuff. From a like practical standpoint, like for us, our research study lab will be doing that. Elise, I presume that they have access to their training, or there's some training that we're going to do with the CRC lab.
Yeah, if they have specific training they need to view, I can send that to them. Otherwise, they're just following the lab manual.
Cool. Next slide. Did we yeah, so purple goes to Maryland, blue goes to the company Cirrus.
Right. So the sera will be aliquoted and put in the cryo vials and then frozen as per the slide before, and then they can stay frozen till the end of the study, and we can just batch send them to Maryland and to Cirrus, and I will manage the shipping containers and send all of that, and then we'll just use our FedEx to try to make that as easy as possible for shipping.
You mean under the study for that patient, right?
Correct.
Yeah.
Actually, no, they could stay longer if you wanted to, as opposed to shipping just a few cryo vials. up to the, how your lab likes to manage theirs. I don't have a specified timeline on that because, if you have a space to keep them frozen in your freezer, we can batch send them.
I suspect we should probably, like, this happens infrequently enough that I think we should send them at the end of each
Yeah.
patient's
Can
Right.
absolutely do
study
that.
period.
Sure.
Okay, and I just want to introduce ourselves. My name is Andrea, and I'm the manager of the team that will be working on the research side of it. And so we will be receiving the lab kits, and then when a patient is enrolled, we'll be, I believe, and we'll work this out with Dr. Biena that we'll be bringing the lab kits over, or that we'll be storing the lab kits at the CRC. Is that right, Elise?
Well, I would think that that we should probably keep at least one patient's worth on the unit, because if this happens in the dead of night, which, but it feels like it's likely to, then we just need to make sure that we don't become late, in arrears on our admission labs, for example.
Gotcha.
And my thinking would be, and Dr. Cancio, since you guys have actually done this, you tell me if this makes sense, but my thinking would be that the entire study's worth of labs would be available at the patient's bedside right at the beginning.
Okay, and I believe we did talk about leaving or having the kits in the storage.
Right, and from a
Okay.
clinical faculty standpoint, like bedside faculty and nursing standpoint, I want to make the burden of effort minimal. I want things to kind of be at their fingertips.
Right.
Cool.
Okay, thank you.
This is just an overview slide about the timing of blood collection, as well as additional data that's going to be collected. Red
And
Cat.
I'm sorry to keep
Yep.
pausing this, but I just want to make sure we get to the questions as we get there. But in terms of SOFA scoring, we agreed that we were going to allow for the presumption of normal bilirubin unless we had specific evidence, because otherwise that would be another study lab. And generally speaking, bilirubin is the thing that you wouldn't routinely have as a part of a SOFA score if you weren't looking for it.
Yeah, I recall that. Yes.
That is correct. Yes. That is correct. Kara McMullen is on, and she'll go through the red cap portion.
Yeah. Hi, everyone. My name is Kara. If you ever have any questions about the database at any time, just let me know. We do have our own RedCap instance. So I've had a couple questions about it, but it is a separate user account than any other RedCap user accounts you might already have. So if you're getting a notification to create a new RedCap account, that's correct. It's a new RedCap account specifically for our burn data center. And there's the link right there. Next slide. So RedCap itself has training videos. If you're new to it, I would recommend doing at least the brief overview and the data entry overview. It's pretty handy, and RedCap's out of Vanderbilt, so I'm sure that you all are familiar with it, but just in case you haven't worked with it before, I would recommend those. Next slide. So we have 3 separate databases in our RedCap instance for this study. We have the main database for data entry. We also have a plasma tracking database. Cirrus tracks the plasma shipped, and then the enrolling center tracks plasma received and how it's distributed. And then there's also a shipment tracking database for tracking that specimen shipping we were just talking about. We don't have any participant tracking databases. That's something that you all will need to do at your local level. There is an Excel template, but we aren't approved to manage any PHI or contact information or anything like that. So that's something that you'll have to do yourselves.
We already have that set up.
Awesome.
Yes. And it actually, our RedCap people called your RedCap people, and we've put links in ours that link straight to yours.
Cool. That's great.
Yeah.
Okay, next slide. So when you go into RedCap, the record ID convention for PROPOLIS is site, _ID number. And we have a lot of customizations in our REDCap. That's one reason we have our own instance. And a lot of those customizations hinge on that ID being correct. So you all at VU are 4, so it'll be 4, your first patient will be 4_1, or you can use like 1001, you can do whatever you want on the other end, but make sure that you have that 4_1. Another note here is that we do want to enter demographic and inclusion and exclusion criteria for every patient screened, even if they end up not consenting or not being eligible in the end for whatever reason. We want to make sure that we can describe the sample of eligible patients. So there's training on all of this on the website, but here's just a screenshot to kind of give you the quick and dirty view of what it looks like to enter a new participant. Next slide. And then because we have these multiple time points and each time point has a decent amount of data within it, we have a grid that comes up, and this looks like regular REDCap, but then we also have the time points, so you can select admission, 12 hours, 24 hours, etc. So you can pivot to whichever time point you want to access for each record using that drop down. And then when you enter new information, you'll start with admission and go through those demographics, injury information, inclusion and exclusion. And then consent is the form where you would do your e-consent if that's what you're using for a particular patient. And then the randomization will randomize them. One thing that did come up, I think, with a previous patient at a different center was that the LAR went through the consent but didn't actually hit submit. So the study staff had to go back and remind them to hit submit, and it was just like an extra step that was, you know, I don't think it caused a huge issue, but it was an extra step. So one thing you can do when people are filling out the e-consent is remind them that they need to read through all the pages and that it's important for them to hit submit at the end. And if you do that up front, hopefully people will do that without having to remind them. Next slide. So this is what the plasma tracking database looks like. It includes information for SEERs to enter when they note the shipment, and then also for blood bank personnel to know about the plasma units. So there's also a training document about this and information about how to find the name for each plasma unit that we're using for tracking on the website.
And one note about this is for the blood bank folks, we're really just wanting to know if the plasma unit was distributed. We don't need to know who it went to in this system. It's primarily being used to track your plasma volumes, really. That's it, not what you're giving the patient, but what you actually have on hand. So that it would be a trigger to reorder, to get with the plasma provider, et cetera.
Next slide. Is there another slide, Monica, for the database? I can't remember. Yeah, so we have on the website, we have several videos and several also training, kind of quick and easy PDFs to read through. Things like data entry, doing e-consent, randomizing, things like that. But does anyone have any questions right now? Okay. I think that's it for me.
I think so. So in regards to safety reporting, it will all be done or conducted through REDCap. So those AE logs and protocol deviations, all of those already exist in REDCap, and you saw them on the screen on the dashboard there. And then all of the assessments and stuff will also be routed through REDCap. So providers at the sites will get automatic emails when an AE has been entered into the system, et cetera. As stated before, we know that this study population is expected to have unrelated serious adverse events, including that list, because they're a sick population, so that we expect to see those. There are some unexpected serious events that are also possible, such as trolley, which is an endpoint of interest. So we'll be tracking it as an endpoint, not as an adverse event. transfusion reactions, thromboembolic events, all of the rest of these are endpoints of interest. So there's no reason to do another safety review when that occurs. That's already going to be in the system. These events are going to be reportable during the first 48 hours post burn, because that's the time frame that was determined for that for this study. The AEs will be identified by the research staff and entered into the log in REDCap, but site PIs are responsible for the initial classification, as is often done, for severity, expected, unexpected, and related. The AEs that are deemed to be severe, unexpected, and related will be considered unanticipated problems and go through prompt reporting. There's a safety monitoring manual that's on the website, and I've sent that out, so that will help with how we're going to report things and what needs to be included in each report and the process flow of that. Unrelated adverse events, whether they're expected or unexpected, will not be recorded on the ECRF. We're only interested in the study-related adverse events.
And then I know you have this, you said you have some like guidance available on this.
I do.
Can we just give an example of what like an unrelated, like expected would be something like, you know, what Steve said earlier about like AKI in a burn, in a like large burn.
Yes.
What, I mean, can we posit something that we would say is like related and severe?
That's,
I can't even really specifically think of
Yeah.
it in that, like, this is not an investigational new drug, right? Like this
correct. It is
is, for all
not.
intents and purposes, it's the same plasma. Like it's not literally the same plasma we'd be giving otherwise, but it is theoretically like something.
Monica, can you go back one?
Correct. Yes.
Go back one. Go back again. So unexpected, serious adverse event.
Right.
You know, these, so trolley, by the way, the risk is extremely low. However, you know, it could be related to the study, and therefore it would be reportable. You know,
And it is reportable. Yeah, and it is reportable. I mean, we're already assessing for it on every patient because it's a secondary endpoint.
Right. I mean, we've tried to anticipate. I don't know if anticipate is the right word. We have tried to think ahead of time what could possibly be related to use of plasma. All right. And they're listed here. Trolley, VTE, or transfusion reaction. You know, what else could happen? I don't know, maybe the patient could get plasma that had bacteria in it. And that would be a new one, but it could happen.
That would absolutely be reportable.
Right.
But again, this is events in the first 48 hours, right?
Correct.
Okay.
Yeah.
Okay.
We tried to really think through the safety monitoring because this is such a data heavy study anyway, and really tried to be, I mean, certainly inclusive. We're not sacrificing patient safety, but not have just try to be judicious with what we were really looking at and the time frame. Certainly, if something happens again, if a patient has a anaphylaxis reaction or something else, that will be an unanticipated problem. It will be severe, unrelated or related, and it will definitely hit the trigger for unanticipated problem. That this patient population is going to have a lot of events, and a lot of them are expected, and there's really, I'm not sure there's value added to making everybody go through the whole AE process for something that is so relatively common in this patient population. This is just a schematic as to deciding when it meets the unanticipated problem trigger, which would be the prompt reporting within, several days that you find it. Again, this is all in the monitoring book, and I can reproduce any of them. I think our bigger challenge is actually going to be in protocol deviations, because that's where we're going to really any of the deviations are probably going to occur with the resuscitation algorithm, right? That's where sort of things are going to become problematic potentially. All protocol deviations will be assessed, and there is a protocol deviation log in REDCap. Certainly protocol deviations that harm a participant or place a participant at risk of harm will automatically be reported as unanticipated problems, which would then meet the prompt reporting. I have just examples of protocol deviations, but certainly there could be something that I haven't anticipated or thought of. I really believe it's going to be the protocol and the consent or possibly enrollment. Those are probably going to be the three biggest areas of issue. Again,
Can we
I
talk
didn't
about
want to.
the last one under protocol, research blood specimen not drawn on time?
Yes.
Like what is the like grace time around that, right? Like it's not going to be to the minute, not going to be that exact, but I'm presuming an hour off is too much. So like
Correct.
what are we treating as like on time?
I'd have to go back to the data collection, but I think it's a plus minus an hour.
Oh, is it plus or minus an hour?
I think so. I think we addressed that, but I will confirm and send that out. I don't, it just came up as a question earlier in the day as well, and I haven't gotten a chance to look at it, but there is a plus minus on that time, knowing that no one's going to stand there and get it at exactly that minute. Yeah.
That sounds right to me, Monica. I think I'm
Right.
remembering an hour as well.
I think so. I think it's plus minus an hour. The clinical monitoring strategy for this study was really used a risk-based strategy. So we looked at things that were high risk to the patient or to the conduct of the study, and we employed some mitigation strategies and then monitoring for that. We will monitor centrally predominantly, and then only do on-site if we really need to, if we're having issues and we need to come to see. But most of the monitoring can be done centrally and virtually. The items that are believed to be critical, data and study procedure, be informed consent, of course, adherence to the protocol eligibility criteria, documenting appropriate accountability and administration of the investigational product, which is not overly cumbersome, and then conduct of the study procedures and assessments. So assessing for study endpoints, which goes to the SOFA calculation each day, and making sure that we have that done, and assuming the normal bilirubin, some protocol required safety assessments, and evaluating and documenting reporting SAEs. Participant level risks were really identified as non-English speaking. So of course we've got translated consents and things were appropriate by institution. The enrollment timeline will be, again, the biggest challenge. And so mitigation strategies include the e-consent, LARs, and whatever site-specific strategies are being employed to try to get patients enrolled within that timeline. And then confidentiality is always a challenge with patients. And so we've got user-based access and secure patient logs, et cetera, for mitigation strategies for that. Protocol violations and adherence, really the only mitigation strategy we can do for that is training and infographics and things like that, support strategies. Potential recruitment issues, those strategies have to be implemented locally. I can't come up with a central strategy necessarily for that. And I know that your staff has worked on that. And I know ISR worked through some subject identification issues recently to make sure that everybody knew about the patients as they came in, and trying to get that consent within the 8-hour timeline. Safety, of course, would be a trial level risk. Plasma distribution and management, which is why we employed the central tracking. Data collection, mitigation strategies there relate to internal validity within REDCap and ECRF availability and manual procedures and training. Unreliable outcomes assessments. We chose to do raw scores instead of having people calculate scores for that reason, to try to have some consistency there. Research specimen, we've talked about that as well, and really the only mitigation strategy is training in that. And then staff training. I think that one of the biggest challenges to this study, aside from enrollment, is the fact that your patients are most likely going to be few and far between, which means everybody's going to forget what to do between one patient and the next. So a specific training strategy is important, however you're going to manage that, whether that be 3 months, 6 months, whatever that's going to be, needs to be employed, because they will forget. I would forget.
Can I ask a question about that? So
Sure.
there's two numbers that I think have changed from like when I first signed on to the study till now, and I want to make sure I understand where they are. The time window of enrollment, which is to say how many years worth of enrollment each center is doing. The first document says 3, and then I remember there was some question about like, is it 24 months instead of 36? And then the other one is, what is our site-specific enrollment target, like number-wise? I know one initial document said 19, and then there was one of 16.
Right. So the entire original period of performance for this grant is 3 years. And so that couldn't be an enrollment period, because of course you would need to have all the startup. So minimally the enrollment would be 24 months. Now, we already know that we're behind. We're behind for plasma reasons. We're behind for blue top tube reasons. We're behind for all kinds of reasons, and all legitimate. And our science officer is completely aware of our challenges. So we will be submitting a no-cost extension to extend everyone's time and the use of their funds for the next year. But I can't submit that till closer to September, till the end of the period of performance. I can't submit it until 90 days prior to the end of my initial period. So that'll be this summer. The initial 1 year no cost extension is almost automatically approved. So that's not going to be an issue. We just need to all sort of plan for it. The initial documents were sent out with an estimated 19 subjects per site, because we had 5 sites. Then we added UTMB Galveston, which decreased the total number enrolled. Now, UTMB Galveston isn't, and they're not enrolling, and they're not ready. So we will clearly want everyone to enroll as many patients as they possibly can get enrolled within the time frame that we're funding this study. So that's why there's a difference in the documents that you saw.
So theoretically, Galveston, if Galveston couldn't enroll ever, then our target is 19 patients at this site over, like, even accounting for the extensions, it'll be,
Sure.
we should expect to be enrolling for about 2 years.
Correct. That's
Okay.
correct. Yes. The total target enrollment for the study is 94 subjects across whatever number of sites are participating. And I know there was some discussion early on about what if a site enrolled more than 19. I don't know that we're going to run up against that problem.
Well, yeah, I mean, from an absolute number standpoint, if you count every burn, that's less than 5% of our burns. But if you count all of our burns over 20%, I mean, not even considering any of their exclusion criteria, many of which will hit plenty of the exclusion criteria.
Correct.
That's closer to 10% a year.
Yes. Yes, it's a challenging enrollment, no doubt about it, for all kinds of reasons.
Okay.
Yeah. I do have a training grid. I have a training manual and a training grid, and with some sort of some expectations as to who would need what kind of training, from clinical research staff to physicians, blood bank, and then clinical staff. And then I have tools. We've created tools for every occasion, essentially, that can be used by each site in whatever fashion that they want, in addition to anything else that you want to do to make sure that your personnel are fully trained. And then the PROPOLIS webpage has all of the documents and all of the resources. It has the training videos on it. It has every manual, all the schematics, the infographics, everything is there. A link to the database is also there. So anything that is, everything should be available there. And if it is not, let me know. Questions?
I have two sort of brass tacks questions. One of them is, again, like I am, you know, especially the staff that's on this call right now are all of our like provider staff, right? So I'm trying to offload the burden of this study to me as much as possible, and to, you know, not them is really important. So my questions are, in terms of screening, because we do want to talk about, you know, we are trying to capture like who screens as ineligible and why also. Who are we opting not to even screen? Let me give you an example. Patient comes in, you know, they're 12 hours out from their burn. I'm not going to screen that person. Or someone is clearly under 20%. I'm not going to ask anybody to even bother screening. Like, unless they hit the theoretical threshold, we're not going to go through it.
I would agree.
Okay. And then among that, like, we're going to screen, like officially screen everyone and write in why they don't, right? So we're talking about like roughly 50 burns a year that are meeting the TBSA criteria, many of which will fail on like basic exclusion criteria of, you know, is it a chemical burn? Is it someone who's, you know, 66? Is it, you know, whatever? And we, you know, we treat children too. So like those people are going to screen out right away. And then, so I just want to decrease that burden down to like the smallest number possible, so we're not spinning up a bunch of people for no reason. The other one is, so sort of separating out research labs from our standard labs, right? So one of them is like, we get, you know, blood gases and lactates, but we don't necessarily put everyone, we don't put an A line in everyone, but trying to account for 48 hours worth of ABGs and also durable access for the purposes of our research labs. Like, is that what sites have been doing, putting ABGs in anyone that like at least enrolls, or putting in an A line in everyone that is enrollable?
No. Dr. Cancer, you're on mute. Yeah.
Yeah, I agree, Monica. We're definitely not requiring or even recommending A-line insertion for purposes of study blood draws.
Correct. The non-research labs are to be entered as available. In other words, we're not dictating you get a CBC every day. We're not dictating you get a chemistry every 6 hours. And we're certainly not dictating you're getting ABGs on a patient that doesn't require ABGs. And we would never tell you you have to put an A line in a patient you would not normally put an A line in.
But I mean,
So
like, is that correct? Because for the first 48
Mhm.
hours, we are expecting an ABG with a lactate, or like the labs entered in include, you know, the components of an ABG, including lactate. I mean, am I wrong about that? Because at hours, you know, and again, this for the larger burns, we you know, our protocol
have
does this too. But
Sure.
like, you know, if somebody resuscitates down to maintenance, or to, you know, effectively like their end of resuscitation at 24 hours, like those additional labs, then like, how are we managing that? Like, does that person, are we still expected to get a ABG at 48 hours?
No.
As written right now, we are.
I think I'll have to go back, because in the protocol it is if available.
Yeah, I do think we need to double check that, because that was not
So-
my reading of the protocol.
Okay.
We'll clarify that. Thank you.
Yes, absolutely. Yeah, because again, if we dictated that, we would then of course have to, you know, pay for that. If that's not what you normally do, then that's a whole different thing. But I will confirm in the protocol.
Hey, if there's no further questions, I just wanted to thank you for joining us on this call and for your questions. I took some notes. I'll get some clarifying information out to you all as well as to the other centers. I really appreciate your participation in this study. I'm excited to be a part of it, and thanks again.
So yeah, just want to
Yes.
reiterate, like anybody, if you have a question, like now's the time to ask. I guess my one other question to Dr. Cancio and I guess to Elise is like, at the end of this call, are we live? Or I mean, I guess we have to wait for the actual like kits to get here. And we don't have a par of our study plasma, but when are we officially within enrollment?
So once you get the kits, and then once I get back there's a training log for everyone to sign that was trained today, and I'll make sure Alyssa has it. And then I need to verify some of the documents, like the financial disclosure and things like that. I will issue you an actual site activation notification, so you will know when all that paperwork has been received and everything is ready to go. I'll have to look in the system. I thought you had plasma in your blood bank, maybe.
I don't think so. I had a call with
Okay.
blood bank on Monday, and I don't think they've received anything yet.
Okay, I need to
Yeah.
get with your plasma provider then,
Yes.
because
And
they
just-
have stuff to send you.
Okay.
Yeah, Tennessee was the one state that was like ready first to.
Yes, yeah, and I had to put it on hold because, we weren't ready.
Yeah.
So I will make sure that happens, but yeah, there will be an actual activation notification, so you know exactly when you're open for enrollment after we get those last few things tied up.
And just
And then-
internally, Dr. Biana, other than the training log today, everybody will have to sign off their training that's been sent in Viva, and then I'll be able to send the DOA out for signatures, so that'll be required as well.
Yeah, so if you guys are on this call, for the other training, which you either were physical, present for, or you've seen the training video, make sure you sign off that you actually have done the training. There's a link from Elise in your email awaiting it.
And Dr. Biana, this is Andrea, and I'm going to be sending you a message. Kay Sarati, who is on this call, and Sue Summers are going to be your coordinators. So I'm going to send an email for you so that we can work out a time to meet independently and kind of talk about where we want to bring the kits over and store things and things like that. And
Okay.
how you need us to walk you guys through.
Okay, Dr. Wagner, do you have any questions while we have Dr. Cancio's attention? You're muted, but I think you're saying no.
I was searching not able to find the mute button. No, I think you guys have gone through and answered a lot and asked the right questions that we were wondering about. So
Thank you, Dr. Wagner.
Okay, well, if everyone else is ready to drop off, that's great. Monica or Dr. Cancio, I do have one question, but we don't have to have the whole group for that.
Sure.
Okay.
And Elise, you're welcome to stay on too, but this is not something that necessarily pertains to you either. So our study infrastructure is off-site relative to the actual burn center in the Vanderbilt Coordinating Center, which is where Elise Seffel works. My question is because to fill in the gaps, especially around the 24-hour aspect of this care, right? If you're getting labs or, if you're getting admissions 24 hours, then these things are going to happen off of bankers hours, which is basically the hours of the VCC. I'm having to engage with residents and stuff to fill in those gaps. So it's not just me 24 hours a day, 7 days a week for 2 years. So my question to you is, how are we at the product of this? How are we managing authorship